Well, hello. Thank you so much for inviting me to this exciting session. I'm slightly jet lagged. It's a 10-hour difference to travel, but fine. So I'll try to talk about two projects, one about stability and one about change. Both of them inside the hippocampus. Do you see you don't see the cursor. Is there a mouse or? One moment, I can deal with it. Now you see the mouse, yeah. Right? Okay, I can even do a better pointer. Okay. The regions that we're looking at are the hippocampus and the entorhinal cortex, the subiculum. These are the regions we are researching in the lab, recording such things as place cells which can be found in CA1 or CA3 and other types of cells such as grid cells or border cells or head direction cells that are more abundant in regions such as the entorhinal cortex and the subiculum. Today I will discuss two projects. The first one is more about stability and the second one is more about change. Yeah. Okay. Not so the volume is the turn up volume.
You don't want to do that.
Okay, but now there is a big echo. So what do we do about the echo? Should I turn off the volume?
Turn off your speaker.
Okay.
There you go.
Now it's okay? And just do that. Yeah?
Yeah.
Good.
You're good. Sorry.
So, and everything is recording?
Yep.
Okay, good. As Jeff was mentioning in the previous lecture, there is the problem of SLAM. So the problem of SLAM is a problem of simultaneous localization and mapping. And the issue is that when we're trying to reconstruct our position within the environment, there are two things that we have to do. The first thing is that we have to understand where we are. And the second thing is we have to understand what is the map. And this is a circular problem. The reason that it's circular, that if we want to reconstruct the map, we have to know where we are. But if we want to know where we are, we have to know how the map looks like. And this is a problem that is well implemented in robotics. You can solve it. You solve it gradually. You both reconstruct the map and reconstruct your position at the same time. But in our field, in the place field, for many years, there is a lot of work on reconstructing our location or trying to understand how do we know where we are within the environment. And this is done, for example, if you think of an environment in which every such round colored region here represents a place cell, then as the rat is running around the environment, different place cells are active, and then by knowing which place cells were active, you can say something about where the animal was in the environment. But the other problem is the problem of trying to understand how the map is constructed by looking at brain recordings. And this is a harder problem, and there's less done about this in the literature. And this is what we try to do in the first project that I will present today. The project was mostly done by Shai Abramson in collaboration with Daniel Zur. Daniel Zur is here in the audience. Daniel, where are you? Up here. So he will know to answer some of the questions better than me. And in collaboration with Hadass Benisti, Ganela Morris, and others. And so what we did is we used neuropixel probes to record from a bunch of regions inside I have to change my mouse to a regular mouse. Sorry about this. Okay. You still see this, right? So we're recording in mice. Thanks for asking. And this is NeuroPixel's recording. So we can record hundreds of nerve cells at the same time. And the question, we have also Shai built this very nice robotic setup in which the camera is following the mouse around inside an environment which we can construct in various shapes. And we record with the neuropixels in these mice. And here you see that what we're doing is what is called a two-room experiments, in which a mouse is running in one room, and you can see the camera and the cable following the mouse. And then after a certain while, the mouse is moving to the other room. As you'll see here, the door opens miraculously, and the mouse is moving to the second room. And we were playing with many shapes of rooms. You can see here various examples of two-room experiments. Yeah. The rooms have various characteristics. I mean, sometimes the separation is more or less horizontal between the rooms. Sometimes it's more vertical, meaning the one room is on the left of the other. There are various shapes. Sometimes you see rooms that are similar to each other, as you see down here on the right, and so on. So we played with various rooms. And the idea was to try to ask how similar is the representation of one room and the other room when doing recordings mostly in the subiculum, although we also did recordings in the entorhinal cortex. And the experiment went like this. The mouse was running 20 minutes in room A, then it was running 20 minutes in room B, and then it was returning to 20 minutes back to room A. And here are examples of trajectories of mice in the two rooms, yeah, in green and orange and in blue, as you can see here. And we recorded hundreds of cells during these experiments. Here are two examples from recordings. The top one is from two rooms which are relatively mirror images of one another. As you can see, this is room A and this is room B down here. And in this example, you see here, this is a border cell that is active along this top wall and left wall over here. And then on the other room, it's active along here on the right wall and the bottom wall over here. So you see this really looks a bit like a mirror image. But of course, we recorded many cells that would not do this exactly. And on the bottom, you see a second example in which, for example, this border cell in the subiculum moved from representing this wall over here to representing this wall over here. But we wanted to do it in a much more systematic way. So what we did is we looked at all of our recordings, and this is a bit of a hard slide. And once we have a population vector of activity of the cells in one recording and another population vector of the cells of the activity in the recording in the other room, we can correlate the population vector in one room to the other room. And what we're showing here are mappings. I'll go for an example here. Okay, let's take an example. So this is what you see here is room A, recording in room A, and then recording in room B. In room A, the recording was twice. So we're showing the first recording always on the left and the second recording on the right. And the color code, every time on the top figure, there's always a pair of figures. On the top figure, it's color coding the x-axis in blue, from blue to red. And on the bottom figure, it's color coding the y-axis from red to blue. And then you can see the transformation, right? So let's say in this example here, the blue triangle, let's say this blue band transformed over here, meaning every point is finding the most similar point in population vector between the two rooms, right? So we can start looking at the transformation of coordinates between rooms. I'll show you here another example. Look at this one over here. So here you see that it's really like a mirror image, like we saw in the single cell examples, right? Because this red band here transforms to this red band here, this blue region here transforms to this blue region here, and so on. But we got, I'll show you, we also applied another method to try to do this mapping, which yielded much more clean results, and I want to focus on this second method. So the second method was doing an encoder to try to connect between the activity of the cells and their representation in the or their position within the environment. And this encoder had two stages. We first built a latent space using an LSTM network, and then a second multi-layer perceptron for transforming the latent space into a position within the environment. And then it looks something like that. So here we're encoding, we have a recording of cells, and at every moment we're encoding from that the position in room A. As you see here, the actual position of the mouse is shown in black, and the reconstructed position is shown in blue here. And then we can also reconstruct a position in room B and the position in room A the second time. And this allows us to build a transformation or the coordinate transformation between the rooms. And then by doing that, we got a much nicer coordinate transformations. It's easier to see here very nicely. Again, if looking at this example, you can see from this experiment the mirror image very nicely, yeah, because here you see that this blue band here transforms to this blue band here, but at the same time the top wall here transforms to this bottom wall down here. So in this example, it's a mirror image. In this example, it's almost an exact copy of the coordinates from room A to room B. In the example here on the right, you see that it's a slight rotation. You see that the coordinates are slightly rotating. But what we noticed when looking at all of these examples is that in many, many cases, the transformations were very, very simple. Although the walls could be very, very complex in shape, still usually we got what is called an affine transformation of the coordinates. Meaning when I say affine transformation, I mean rotation, reflection, scaling, shearing, and translation. So in our hands, in many, many environments and many, many mice, we got in many, many cases an affine transformation. Here, there's a bit of a quantification. I'll just show an example here. So this is when we looked at, this is how well the network was predicting position. This is in a room when taking all of the neurons. These were when we were taking only the entorhinal cortex neurons or the subiculum neurons, and these are when we were taking specific cell type, allocentric boundary cells, egocentric boundary cells, head direction to check which classes of neurons, subclasses were doing the prediction better. And also, we checked how affine was the transformation. So, of course, when transforming from room A to room A, the transformation was usually affine, but also we got a very good affinity score when looking at transformations from room A to room B. I just got the 5-minute thing, so I may, it's 5 minutes including the questions, right? So, or not including, okay. So I'm contemplating if to tell you the second project or to just stop after the first. Okay, okay. This is showing the prediction. I think I'll just talk about one project then, so we have time for questions. So here you're seeing the prediction score from the network on the x-axis and how affine a transformation is on the y-axis. And you see that in many cases, each such a square is one experiment. Okay, and what you see is that for many of the experiments in which the prediction worked very well of the network, then you also got an affine transformation. So it seems that that in many cases we have an affine encoding. Just about the properties of this encoding. It was hard for us to find exactly which transformation occurred. In many cases, we did not manage to find a rule of exactly what was a transformation, except for one case. In the case that there was a reflection, we saw that the transformation of coordinates tended to occur along the reflection axis, which tended to be the boundary between the rooms. So you can see here that, for example, when the boundary between the room is more or less a 90-degree boundary, as you see down here, the reflection tended to be using this axis, and similar in the other cases. So at least for reflection, we seem to have a simple rule for the transformation of coordinates between the rooms. And this is the first project. So we saw an evidence for coordinate transformation of border cells from egocentric to world-based coordinates, and preliminary results demonstrating that border cells in two rooms map one onto another through an affine transformation, meaning rotation, translation, extraction, expansion, or contraction. And the second project is published, so then you can read about it yourself. So I'll just say that it's about hippocampal drift, and it was published in Neuroscience, so go and have a look. It's an interesting work as well, but I will skip it due to the lack of time, and we'll go directly to the thank you slide.
You have 2
2.5 minutes to say something about this project. Let's do that. Okay. So, what we did is we did calcium imaging in the hippocampus, and we looked at the drift. And the question we were asking is whether representational drift is a function of time or is it a function of experience? And we did an experiment to check that. So the animal was running 10 minutes in environment A along linear track A, and then it was running more than 2 hours in environment B, and then it was running 10 minutes in A and 10 minutes in B. And we looked at the comparison between the recording in the first A and the second A, and the recording in the beginning of B and the second B. And the idea is that from the first A to the second A and from the first B to the second B, it's the same time, but a very, very different experience. And when you look at the cells as a function in these experiments, as a function of where they fired along the trajectory, so the x-axis here is the position in the trajectory, the y-axis is the places sorted according to where they fire. And you see that if you compare the first time in A and the second time in A, you see it's the same map. Well, if you compare the first time in B and the second time in B, you see that it's very different, suggesting that when the animal was more time in the environment, let's say in environment A, it was 10 to 20 minutes in total, while in environment B, it was more than 2 hours in total, you got that when there was more experience in the environment, then there was a faster drift, or there was more drift. And that's the basic result. We did also an opposite control in which the animals were a similar time in both environments, but the time between the first time in environment B and the second time in environment B was much longer. In this case, when the time changed, but the experience didn't, you see that there was no remapping. So if you look at, or there was less drift. If you compare the first time in A to the second time in A, here you see that it's very similar, while in the first example I showed you, you see that the first time in A, the second time in A, it's similar, but there was much more drift from the first time in B and the second time in B, and this is just a quantification of that. Yeah, so that was my 2 minutes version of the second project. So what we see in the second project is that representational drift is dependent on experience in context and not time, and active experience seems to be the main determinant. So thank you very much. Yeah.
James from downtown, please. Result about that super explanation stands to background. And so what does that mean about remote?
Yeah, I don't think it explains you can place cells. I think in the case of place cells, things are much less clear. I think the remapping the remapping rules for place cells are less clear than the remapping rules for subicular cells like the ones we were seeing here. Yeah.
Second, sense of whether they're dependent on anything. Showed one example where the animal, you opened up the secret hatchway in the bottom. What if you did it in the top? Would that have an impact? Do you have any sense of that?
Yeah, I think it's a very good point. We haven't checked it systematically, and Daniel is here to write it down that we need to look at it. Okay, thanks. Yeah.
You know how much of the acting result is due to like actually similar to the mapping of points into space and how much of that is playing with research destroy?
The question is how much is the final result of smoothness? Well, at least our network is not taking the smoothness into account, because our network is doing the encoding from a space time, from the activity and not directly from the rate maps, right? So it's more systems. I'm wondering if you expect a smoothness to lead to affinity. I'm not sure. I don't think that you must expect that, right? Coordinate transformations can be much more random in shape, right? And much less regular. But it's a good point that I think we will have to think about. Thanks. Yeah.
I feel that after the LSTN, have you tried putting more linear layers? So it might capture some of the non-affine transport.
Yeah, we see a lot of cases. I mean, when you just check randomly, when you look at simulations, we see many examples of which the transformation is not affine. It's not happening by default. In most cases, the transformation does not come affine. What's nice is that in the real data, we see an affine in tendency for affine transformation. Thank you very much.